Okay, so we're going to cover chapter 12. We're going to actually break chapter 12 up into two pieces, as you can see from the vocab list and the chapter outline. And that's because there's just a lot of information in chapter 12. So in part 1, we're going to cover Mendelian genetics. We're going to talk about both the mono and dihybrid crosses, so looking at one trait or two traits at the same time. And then in chapter 12, part 2, we're going to look at all of the exceptions to Mendelian rules. Okay, so the chapter in the textbook is not really broken up that way. So you may have to kind of jump around and look for stuff as you're filling out the vocab and chapter outlines, but that's how I like to divide it. There's going to be a little bit of information in chapter 12, part 2 that's actually in chapter 13. I don't ask you to outline that or really talk about that too much in the vocab list, but we will cover it in the lecture. It has to do with linkage, okay, and genetic maps. So we'll get to that in part 2. So just to let you know, I know the chapter doesn't go totally perfectly with what we have in the chapter outline, but we'll hopefully made the best of it. Okay, so we're going to start off again in part 1 talking about Gregor Mendel. So let me move my camera so you can see his picture. So first off, again, we're studying genetics. This is nice. We're going to spend this week and next week in lecture, and you actually spend a couple of weeks in lab on genetics, so you should have a good understanding of it by the time we're finished. Genetics is the study of heredity. It's the study of the transmission of genetic information, traits, from parent to offspring, or from one generation to the next. And in chapter 11 and in lab last week, we talked about meiosis. And meiosis is crucial to understanding genetics. You have to really understand that process of meiosis to understand how genetic information is passed from parent to offspring, right? In sexually reproducing diploid organisms, like us and like corn, okay, or fruit flies. We're going to be looking at a bunch of genetic problems this week and next week. And everything that we're going to look at is diploid organisms. So again, hopefully you know what that means. A diploid organism is going to be an organism that inherits 2 alleles for every gene. Okay, and we're going to go back over the definition of gene and alleles. Actually, we went over those definitions in the last chapter, and we'll talk more about those definitions in this chapter as well. Hopefully you know that a gene is a location on a chromosome, and that organisms like us, diploid organisms, we inherit 2 alleles. So 2 genes, 2 alleles. Those alleles might be the same, or they might be 2 different alleles. And that's going to tell us what we have for those traits. Remember, you inherited one of those alleles from your mom, and one of those alleles from your dad. Okay, and in your mom and your dad, meiosis occurred to produce gametes that contained those alleles. So everything there is related. Okay, so again, the definition of genetics is the study of inheritance, the study of heredity, the transmission of genetic information from one generation to the next. Now, the father of genetics, the scientist who gets the credit for the work, which he did a lot of work on this, so rightfully so, was Gregor Mendel. Okay, so you probably have heard of Mendel. Again, we consider him to be the father of genetics, which means that he, did all of the work that led to us knowing there's going to be 2 laws here. Very interestingly, Gregor Mendel did his work before we knew about meiosis, before we knew about chromosomes, before we really knew what genes were or what alleles were. So he was kind of working in the dark. Which is brilliant that he was able to do everything that he did without really knowing too much information. So that's pretty great. Okay, so I'll move my picture here. I like to show you guys my face so you feel like we're in the same room, but then it gets in the way of some things. So again, Gregor Mendel, his work was done again in the dark, and it was brilliant. And the reason why he was really able to figure out all that he did was he picked the perfect model organism. Okay, what is that? That's the organism that he chose to study. He chose the common garden pea plant, which is pictured up there on the top. Okay, and in the garden pea plant, he chose to study traits that varied in an either or fashion. Okay, this is really, really important. Flower color was either purple or white, nothing else. Pea shape was either wrinkled or smooth. Okay, pea color was either green or yellow. So all of the traits that he chose to look at varied in an either or fashion. There wasn't a third or fourth potential phenotype. We'll get to phenotype in just a second. The second thing that made his system really unique and great to work with is that he could control breeding really, really easily. And we'll talk about how he did that. Okay, when he went to breed, he would breed specific organisms, and that's pretty easy to do with plants, or with fruit flies, or even with mice in the lab setting. However, pea plants, they produce multiple offspring in one crossing, which is nice, just like the corn that we're studying in lab. So again, great model organism, easy to grow. Scientists work with fruit flies too when we talk about genetics. Fruit flies gets a little bit messier. You can end up with an infestation. I remember in a genetics lab that I had in college, we had fruit flies everywhere. That was our model organism. They're pretty easy to breed. They're pretty easy to house. They're small. They have just a few chromosomes, so they're great for genetic studies. But Mendel chose the pea plant. Okay. He used the scientific method. He conducted these experiments that were well thought out and well planned. He collected quantitative data, right? So numbers, right, to analyze from really large sample sizes. And he repeated his experiments over and over and over again. Okay. So, they were repeatable, which is what we want in the sciences. Through his work, we ended up with 2 laws of heredity that still hold true today. So what were those laws? The law of segregation, or we could use the term principle as well. And the second one was the law of independent assortment. And again, both of these lead back to meiosis. So you got to make sure you understood chapter 11. So before we go any further, let's just make sure that we understand some terms. We talked about chromosomes in the last chapter, so you know that chromosomes are condensed DNA that contain genes. You know that a gene is a specific sequence, a location on a chromosome that codes for a specific trait or characteristic. You know what homologous chromosomes are, right? Those are chromosome pairs that are found in diploid organisms like us. An organism is going to inherit 2 copies of each chromosome, or 2 chromosomes making a homologous pair. And each of those chromosomes will carry a form of the gene called an allele. Okay? Now here's some new terms. If the organism inherits alleles that are the same for a particular gene, we say that the organism is homozygous for that gene. So let's say, for example, both your mom and your dad give you the big E for eye color, right? We're going to look in lab this week. You're going to find out that having pigmented eyes is dominant to having non-pigmented eyes, which in our terms, let's say brown eyes is dominant to blue eyes. Okay, so if both your mom and your dad create gametes through meiosis that contain the dominant allele, the big E allele, then you will end up being big E, big E. And so you would be homozygous for the eye color gene. So big E, it's hard to write on the computer. Let me use my finger. Big E, that's going to be homozygous. It'd be homozygous dominant. Okay. If both of your parents gave you the recessive allele, the allele that codes for blue eyes, you would be little e, little e. We would say that you are homozygous recessive. Okay, so that term homozygous means you inherited 2 of the same alleles. If you inherit 2 different alleles, so if mom gives you big E, and maybe dad gives you little e, we would say that you are heterozygous for the eye color gene. Okay, so homozygous, the same, heterozygous, two different alleles. And again, in this first part of the chapter 12, part 1, we're going to focus on classical or Mendelian genetics. And then again, in part 2, we're going to look at all of the exceptions to Mendel's rules. Okay, so a little bit more about Gregor Mendel. He lived from 1882 to 1884. He was a student, a teacher, a scientist, and a monk, actually. And in 1856, he began to do his research using the common garden pea plant. It took him about 10 years to gather all of the data that he gathered. And in 1865, he presented his results to the local Natural History Society, kind of like a modern-day conference, scientific conference. And what he said is that, what he found is that traits are transmitted from parent to offspring, right, independently of other traits. So your gene that codes for eye color is going to be transferred independently of your gene that codes for, the enzyme catalase. Okay, those are two different genes coding for two different things, and they would be transferred independently of each other. Okay, and he also came up with the idea of dominant and recessive. Okay, genes that are dominant are going to give you a specific, we haven't talked about phenotype yet, but they're going to give you a specific, maybe if we're talking about eye color, the dominant allele is going to give you brown eyes. The recessive allele, if it's with the dominant allele, you're still going to have brown eyes. You have to be homozygous recessive to end up with the recessive phenotype, blue eyes. Okay, we'll talk more about that in just a minute. In 1866, after he presented his work, he published his work, right? And the title of his manuscript was Experiments in Plant Hybridization. And at the time, we didn't know, again, about chromosomes or about meiosis or about really too much about genes. And so the scientific community didn't really pick up on his work or accept or use his work until it was rediscovered in the 1900s after he was already deceased, unfortunately. Okay, so let's find a good spot for this. Let's go up there. So up until really the 1900s, scientists and the general community, the prevailing theory of the time for inheritance was called the blending theory of inheritance. So what scientists thought was that following fertilization, parental traits, they get kind of mixed and lost or absorbed. They get blended together, and offspring end up as an intermediate between the two parents. Okay? And so scientists or even the general public, when they're looking at traits, for example, like human height, the blending theory of inheritance kind of fits, right? Or it seems like it fits. It doesn't actually fit, but it seems like it does. You have two parents. One parent's really tall, one parent's on the shorter side, and you end up with children that are kind of in the middle of the two parents. Okay? Traits like human height or even skin color are not great traits to study. Thankfully, Mendel didn't pick those traits because they show what's called continuous variation. Human height is actually, and skin color, are both controlled by multiple genes interacting together to produce a specific outcome. Instead of just one gene, one outcome. So again, studying traits like that gave really false evidence for this blending theory of inheritance. So thankfully, when Mendel chose his model organism and he chose his traits to study, he studied traits that were discontinuous, which means that those traits were controlled by one gene. And when you look at offspring, you do not get this blended phenotype. Okay? So again, Mendel looking at the garden pea plant, let's use one example, flower color. If you had purple and white parents, you ended up with purple offspring. If you took those offspring and you mated them, you ended up with a very specific ratio. It's called the 3-to-1 ratio of purple to white flowers. So everything was really qualitative and predictable. Okay? And that again is because Mendel chose discontinuous traits that varied in an either or fashion that did not seem to get blended in offspring. Okay, so it's all about Mendel's choice of organism and his choice of traits to study. Thankfully, he picked the right stuff. Okay, so again, his model organism is the common garden pea plant. Its Latin name is there for you, right? Let's do some taxonomy. So the genus is Piscium, and the species is Sativum. Okay, remember that from last semester? Every organism on the planet gets a two-part scientific name. The first part of the name is the genus, and the second part is the species. The genus is always capitalized, and both words are always italicized. Or if you're writing them and you can't write italics, you could underline it. Okay, so there's its scientific name. Here is a key thing that, again, Mendel was able to set these up. So if you have these pea plants, you can allow them to self-fertilize, which means that, within a flower of the plant, pollen, which contains sperm, if you remember from last semester when we did those plant dissections, pollen produced by the anther, the male part of the plant, is going to contain sperm, and it can fall onto the pistil, the female part of the plant. The sperm can swim down and fertilize the eggs, all in one flower, okay, which again is called self-fertilization. Some plants can do this. The common garden pea plant can. And through self-fertilization, round after round after round, Mendel saw that he had what are called true breeding or pure plants. So these are plants that generation after generation of self-fertilization, every single plant, every offspring looks the same as the parent. Okay, which is pretty neat. He was able to do this, and he would start all of his studies with true breeding, pure lines, pure plants. Okay? And I'll give you a little bit of a precursor. We're going to cover this in the next slide, but let's say true breeding, let's say we're looking at flower color. True breeding pure plants, if they were producing purple flowers, they would be homozygous dominant, big P, big P. If they were true breeding pure white flower producing plants, they would be little p, little p. And I'm going to put a line over these p's. Whenever I'm doing genetics and I'm using a letter to represent a gene that the capital and lowercase look the same, I always put a line over the lowercase letter. So if I'm using the letter P or the letter W or the letter S, right, the capital and lowercase letter kind of looks the same. So I always make sure, especially if I'm writing, that I really put a line over that lowercase p, because you can get confused and then the result of your genetic crosses can become screwed up. Okay, so again, pure or true breeding plants, if I had a big P plant that produced purple flowers, purple is dominant to white, then generation after generation of self-fertilization would always give me purple flowered plants. Those plants would be homozygous dominant. For the little p, those would be plants that produced white flowers, generation after generation after generation. So they would be true breeding, pure white flowered plants. Their genotype, we're going to get to this terminology, I'm just trying to use it to get you used to it. The genotype, the genetics, what alleles that plant has would be little p, we also could say that that plant is homozygous recessive. Okay? Okay. So, good model system. You can do self-fertilization to get pure, true breeding plants. We're going to call those the parent plants. What else? The common garden pea plant has a short maturation period, so it grows to maturity in one season. Okay, so mating is pretty easy. You could get large quantities of garden peas, I should say could, be cultivated simultaneously. So you have a lot of organisms to study so that you have a large sample size. Okay, easy to breed, easy to house, not messy. Okay, so let's look at Mendel's crosses. Maybe I should get rid of my my video for now, or let's just push it over here to the corner. Okay. So this is what he did. So he started off, again, with parental plants. Okay, so I want you to make sure that you understand this pea generation. The pea generation, he started off with pure or true breeding lines. So again, for this parental generation, we've got flower number 1, which is purple. So if I were to put the genotype, what alleles does this plant have? Big P, big P. The phenotype, I'm using genotype and phenotype. We're going to cover those terms in the next slide, but I know you did them on your vocab list, so you got those, hopefully. So the genotype of the purple flower producing plants is going to be big P, big P. The phenotype is purple. So what the plant looks like is going to be purple. The white, pure parental lines are going to be little p, little p. I'll put lines over those. Okay, so again, you've got 2 parents. They're both true breeding. One is true breeding purple, okay, which would be homozygous dominant, big P, big P. One is true breeding white, which would be homozygous recessive, little p, little p. Okay, here's the key thing that he did. He took the purple parents, and what he did is he removed, in that first figure, he cut with his scissors, he cut out all of the anthers, which are the male part of the flower. The part of the flower that produces pollen, which contains sperm. So he cut those all out. And then what he did is he took a paintbrush and he collected pollen from his white flower producing plants. And he brushed that on to the stigma, the sticky stigma, the female part of the plant. Very carefully brush that on so that the pollen from the white flower producing plants is going to now fertilize the eggs of the purple flower producing plants. So he controlled the mating. Okay, brilliant. He then allowed those purple flowered plants that now have had that pollen put on them, he allowed fertilization to occur, and then he allowed each one of those fertilized eggs to grow into a pea. Okay, so in figure number 4 right here, where we've got our peas, he took each of those peas and he planted them, and he allowed them to grow, and he ended up with what we call the F1 generation. Okay, the F1 generation, that F stands for filial. So it's the first filial generation of offspring. So we started off with parents, generation P, that were all true breeding. He crossed them. We can also call this a hybridization study. He crossed them. And what he ended up with was the first filial generation. Now look at those plants. All of them produce purple flowers. He could have stopped there and said, I don't know. Okay, and that's a lot of scientists of that time said that. They would say that that white gene, or they didn't say gene, the white got lost. It's gone. The offspring, there was blending that happened, blending of fluids, they would say, and that white characteristic is gone. All the plants are purple. So luckily, Mendel didn't stop there. Let's go to the next slide. What he did was he then took 2, this figure is from your book. So we'll back up 1 second. So let's start up here at the parents. So again, he had parents that produced purple flowers, and he had parents that produced white flowers over and over and over again. So these are true breeding parent plants. He mated these in a controlled manner, and what he ended up with was all purple plants. These are called the hybrid progeny. That term progeny just means offspring. All of them have purple flowers. Again, that's the F1 generation. Now here's the key part. He took members of the F1 generation and he then crossed them or allowed them to self-fertilize. Either one is going to give you the same results. And what he ended up with is called the F2 generation. Okay, so the generation after the F1, after you allow them to mate. And he ended up with, okay, so in this case, if he looked at all of the offspring, okay, so after he mated these, he took the peas, he planted them, and he allowed plants to grow. He ended up with 705 plants that produced purple flowers and 224 plants that produced white flowers. Okay, so what's the ratio? So you're going to have to figure out ratios in lab. So let me show you how to do this. You're going to take the lower number, in this case it's 224, and divide it by itself. Okay, so 224 divided by 224 is 1. Okay, because you're trying to look for a ratio of something to 1. And then you're going to take 705 and also divide that by 224. And if you do that math, you should get 3.15 to 1. Okay, that's your ratio of purple to white flowered plants. That's the actual ratio. Okay, which is about a 3 to 1. Okay, you're going to see this 3 to 1 ratio in lab this week. You've got 4 cobs of corn to look at. 2 of them are going to show a 3 to 1 ratio. Okay, think about that. How do you get the 3 to 1 ratio? Let's go a little bit further. So he did these hybridization and then crossing of the hybrids. He did that with more than just flower color. He looked at seed color. He looked at seed shape. He looked at pod shape, pod color, flower position, stem length. He looked at all of these different characteristics in the common garden pea plant, and every single time he got about a 3 to 1 ratio when he crossed the hybrids. So the F2 generation. That's really good evidence that there's something more going on than just blending. Okay? So in 1865, again, he reported these findings at that scientific meeting for all 7 of these different traits that he had been studying over the past 10 years. And he came up with the term trait. He kind of coined that. And that's going to be a variation in the physical appearance of a heritable characteristic. Okay, so flower color would be a trait. Seed color would be a trait. And again, the findings that he was able to mathematically produce were pretty much the same for all 7 traits. So notice that 3 to 1 ratio. You can see the ratios here on the right hand side of this chart. Those are the ratios. Okay, so every time when he collected the data, he got about a 3 to 1 ratio. It didn't matter what trait he was looking at. Okay, so before we go any further, let's make sure we're all together. So I got a couple questions for you. So the first one, Mendel performed hybridizations by transferring pollen from the blank of the male plant to the female ova. What is that structure called in the male plant? Hopefully, anther. So the anther is going to be the male part of the plant. Remember, the anther kind of looks like this. Maybe. Okay. So what I just drew there is called the filament. That's that stalk-like structure. And at the top will be, oh, that's terrible, will be the structure which actually produces the pollen, which is called the anther. This whole thing collectively is called the stamen. Okay. The next question here, imagine you are performing a cross involving seed color and garden pea plants. The what F1 offspring would you expect if you crossed true breeding parents with green seeds with true breeding parents that have yellow seeds? Yellow is dominant over green. Okay, so let's figure this out a little bit. Let me erase our... Hold on. Okay, good. Get my pen back. Okay, so we've got the common garden pea plant, and we're talking about two parents that are true breeding, and yellow is dominant to green. So let's use the letter Y. Okay, you can really choose any letter that you'd like. So yellow seed plants are going to be homozygous dominant, big Y, big Y. And we're going to mate those. We use the little X to represent a mating. We're going to mate those with plants that produced green seeds that were also true breeding. So that's going to be lowercase y, lowercase y. Okay, so if we did that cross, what would the offspring look like? Would all of the offspring be yellow? No. 100% yellow green seeds. Blue, 100% yellow seeds. Green, 50% yellow, 50% green. Or yellow, 25% green, 75% yellow. So hopefully you answered 100% yellow seeds. So this would be the parent generation, the P. So we're looking for the F1 generation, first filial. And this cross that we have up here would produce all hybrid plants. All of those plants would inherit one allele from one parent, the big W, sorry, the capital Y, and all of the offspring would inherit the second allele from the other parent, little y, right? So all F1 offspring are going to be what we call heterozygous, big Y, little y. Okay, hopefully that made sense. Okay. We're going to do lots of genetics practice problems together, so we'll keep writing those out and make sure we got it. So what we know is that blending wasn't happening, that traits were not lost in the F1 generation. Thankfully, since he carried his studies out to the F2 generation and beyond, those traits reappeared. So let's go back to flower color. So remember, so his parent plants were, I forgot what letter we used. I think we used, let's use P. Okay, I can't remember if we use P or R when we look at flower color. So purple parental plants were big P, purple's dominant to white. And the white plants were little p, little p. When we crossed those, we ended up with the F1. And the F1 was all big P, little p. Okay, and when we crossed those, we got an F2 generation. And that F2 generation showed a 3-to-1 ratio of purple to white. And we haven't talked about how yet, and we're going to. So let's make sure we understand some terminology. So when we say dominant trait, in the case of flower color, purple is dominant. That trait is always expressed, whether it's a true breeding purple flowered plant or the heterozygous big P, little p plant. Those plants, so either one of these genotypes, I'm going to circle them, big P, or big P, little p, both of those will give you purple plants because the purple allele, big P, is dominant to the white allele, little p. Okay? The little p, we say, is the recessive allele, or white is a recessive trait. What does that mean? The recessive allele is hidden or masked when you have a heterozygous or hybrid organism. okay? It's masked because the dominant allele is present. So in a plant that is big P, little p, heterozygous, that little p allele does not get expressed. It's masked. It's hidden. Okay? Only plants that are little p will end up being white. Okay? So the key thing here, and again, thanks to Mendel going out to the F2 generation, those recessive traits, they're not lost. So if you look at the F1 generation, the hybrids or the heterozygotes, big P, little p, if Mendel stopped there, he would have went along with the rest of the scientific community and thought, oh, the white trait got lost. It's absorbed. It's gone. Thankfully, he took his studies out to the F2 generation, and that's where the white trait reappeared. So that 3-to-1 ratio, for every one white plant, he ended up with 3 purple plants. But the key thing here is that he ended up with white plants. That trait wasn't gone. It wasn't absorbed. It wasn't lost. It was still there, and it could still be expressed. Okay. So from these studies, what Mendel proposed is that plants, or any diploid organism, has 2 copies of each trait. So plants inherit 2 copies of the flower color trait. Okay? Now we say that these pea plants inherited 2 alleles for every gene, but again, that terminology didn't exist. So Mendel actually said they just get 2, he actually called them unit factors. Okay? Where do these 2 copies come from? Well, they come from the parents. So each parent is going to transmit 1 of its 2 copies to its offspring during fertilization. How did that happen in the parent? Well, meiosis. So meiosis occurred in the parent organism, and meiosis produces gametes that only have 1 allele for each gene. Remember from last chapter in meiosis, we know that gametes, egg and sperm cells, are haploid. They only contain 1 allele for each gene. The resulting zygote or offspring gets 2 copies, 1 from mom, 1 from dad, or 1 allele from mom and 1 allele from dad. Okay, so again, these white flowered plants, you would have had to inherit 2 recessive alleles. Purple flowered plants is either this, big P, or big P, little p. And that's because purple is dominant to white. So all of the white flowered plants, we would say, are homozygous recessive. The purple flowered plants are either homozygous dominant or heterozygous. Okay, so here's some more terms. So again, diploid organisms like us, or the corn that you're going to study in lab, or the flowering plants that, the pea plants that Mendel studied, we are all diploid. We inherit 2 alleles for every trait. Now we are going to study genetics in a haploid organism. So fungi, the fungus that we're going to study, hopefully, is called Sordaria formicola. It's a fungus that actually is haploid most of its life cycle. It's a haploid dominant organism. And so when we study genetics in that organism, they only inherit 1 allele for each trait, and that allele gets expressed. So it's actually much easier to study genetics in fungi than it is to study it in humans or in corn or in flowering plants. Okay, so we are diploid, they're haploid. So some terms that I've been throwing around that hopefully you understand, but here they are just in case. Phenotype. A phenotype is a physical expression of a trait. It's what gets observed. So when I say purple flowers or white flowers or brown eyes or blue eyes, or in lab, we're going to talk about corn kernel color, so purple and white kernels, or wrinkled or smooth kernels. Those are all phenotypes, what it actually looks like. The genotype is different. It's the actual alleles that are present that make up the genetic makeup of the organism. Okay? And so I've been doing this already. We demonstrate alleles using one or two letters, and I like to just use one letter, so I'm going to actually change this. I forgot to change it on our PowerPoint, but I'll just write it. So let's look at flower color, right? So you can see right here for flower color, we've been using a big P to represent purple and a lowercase p to represent white. And again, because P looks the same in the capital and lowercase, I would have, I like to put a line over my lowercase recessive letter. In lab, you're going to look at kernel color, right? And so for purple kernels, you're going to use the capital letter R. And for yellow kernels, you're going to use the lowercase letter r. So on Cobb's 1 and 3, no, Cobb's 1 and 2 this week, you're going to be looking at the kernel phenotypes. And so for genotype, again, you're going to use big R to represent the purple allele, and you're going to use little r to represent the yellow allele. And so kernels in lab that are purple. Okay, let's do purple first. So purple kernels can be produced by either big R. So if two dominant alleles are inherited, then the kernel is going to be purple. Or big R, little r. Okay. I didn't need to put that line there, but it's a habit. So the heterozygous condition will also give you purple kernels. Yellow kernels are only produced by little r. Okay, so over here, these are genotypes. Okay, the top two would give you the phenotype purple, and little r would give you the phenotype yellow. Okay, students sometimes get a little bit confused there. So you want to make sure that you write the proper, if it's phenotype, you're going to write purple or yellow. If it's genotype, then you're going to give me the alleles that are present. Okay, let's go on to the second trait that you're going to study in lab this week. So Cobb's 3 and 4, you're going to be looking at the shape of the kernel. And so kernels can either be starchy, which causes this smooth appearance, or kernels can be wrinkled, which causes them to look shriveled up. Okay, in your lab book, I haven't changed this yet. I'm going to in the next edition. Your lab book tells you to use SU as the allele. Just use the letter S. Let's make life a little bit simpler. The U being there confuses students sometimes. So for starchy, get rid of this. You're just going to use S, and the same thing for sweet. You're going to use S. And starchy is dominant to sweet, or we could say smooth is dominant to wrinkled. And so for your smooth starchy alleles, you're going to have the capital letter S. And for your wrinkled sweet alleles, you're going to have a lowercase letter s. Okay, so if you have a starchy kernel, its genotype is either big S, or big S, little s. Both of those genotypes are going to give you the starchy phenotype. Now your wrinkled or sweet kernels, they're going to have the genotype little s. They're going to be homozygous recessive. Okay? Okay. So genotype versus phenotype. Okay, so I've been using those terms, homozygous dominant, homozygous recessive. Let's put them on paper for us. So again, a homozygous organism is going to be one that inherits two of the same alleles for a given gene. So if we're talking in lab about kernel color, if we've got a purple kernel, that kernel could be big R, homozygous dominant. If we have, let's jump down to, if we have yellow kernels, those would be little r, homozygous recessive. So again, the term homozygous tells you two of the same alleles. If they're two dominant alleles, it's going to be a homozygous dominant. If it's two recessive alleles, it's going to be a homozygous recessive. Let's do flower color. So purple flowered plants could be big P, homozygous dominant. White flowered plants will be little p, homozygous recessive. Heterozygotes are going to be organisms that inherit two different alleles for a given gene. So again, in lab, our purple kernels are going to be, can be big R, little r, and the purple flower phenotype can be conferred by big P, little p. Remember, I'm saying can because the homozygous dominant and the heterozygote both have the same phenotype. Okay. Okay, let's quickly do kernel shape because that's what you're doing in lab. So again, homozygous starchy or smooth would be big S, big S. Okay, that would be the homozygous dominant. And the homozygous recessive would be little s, and those would be wrinkled. The heterozygote would be big S, little s. That would be the genotype. The phenotype would be starchy or smooth. Okay, so you want to make sure you can use the terms genotype and phenotype, and that you can also explain what the genotype means. So is it a heterozygote? Is it a homozygous? And is it dominant or recessive? So the terms are important. Okay, so let's do some Punnett squares. We haven't done this yet. So let me move this over here. Okay, so again, we're still working with the common garden pea plant, but now we're looking at pea color. Okay, and so peas can either be yellow or green, and yellow is dominant to green. Now, we're studying the transmission on this slide of just pea color, which means we're looking at just one genetic characteristic, one trait. So we're going to call this a monohybrid cross. So mono meaning one. We're studying just one gene here. Okay, in a little while, in the second part of the chapter, we're going to study two genes at the same time. And in lab next week, you're going to study two genes at the same time. And what those are called are dihybrid crosses. So mono means one, di means two. Okay, so let's start with this monohybrid cross. So again, we're looking at color. And we know that yellow is dominant to green. And so right here, we've defined our alleles. So big Y is going to be yellow, and little y is going to be green. Let's make sure we understand what genotypes can be produced. So big Y would be homozygous yellow. Big Y, little y would be heterozygous, still giving you yellow. And little y would be homozygous green. Okay, so let's start at the top of our figure here. So notice we've got the P, which is the parental generation. That tells you that the plants being crossed here are true breeding pure plants. So the big Y yellow plant is always going to produce yellow peas. And so when meiosis occurs in this organism, the only type of gametes that this organism can possibly form are gametes that contain the dominant allele. That's all that organism has. Same thing if we come over here to the green pea producing plants. These are true breeding parents. So the only type or the only color pea that's being produced are green peas. And so when meiosis occurs in this individual, again, the only gamete that could possibly be produced are little y gametes, the recessive allele. Okay, so now you've got gametes here coming together. So let's put a little plus sign. This is fertilization. All of the offspring are going to have this genotype. So again, these are the F1s. They're all going to be heterozygous, or we could also use the term hybrids. Okay, so if we look at these plants, they're all going to have the genotype big Y, little y, heterozygous. And they're all going to produce yellow peas. Okay? So now what if we cross two of these plants? So again, take pollen from one, put it on the female reproductive structure of the other, or just allow self-fertilization, what's going to happen? Well, first we have to think about what kind of gametes this plant will produce. Half of its gametes will contain the big Y, the dominant allele, and half of its gametes are going to contain the little y allele. Okay, this gets a little bit more complicated. Now we need to set up something called the Punnett square. So what a Punnett square is it's going to be kind of a graphical representation of this genetic cross. And that Punnett square is shown right here. Okay, and on the side, the left-hand side here, we're showing the gametes of one parent. So one parent, we're going to show 50% or half of its gametes will be big Y, and half of its gametes are going to contain the little y allele. The other parent we're going to put across the top. Again, 50% of its gametes contain the dominant allele, big Y, and 50% of its gametes contain the little y, recessive allele. Now inside the box, you show the result of fertilization. So in this first box here, you're saying, okay, so if this gamete fertilizes this gamete, this is what I get. So if a gamete containing the big Y is fertilized by a second gamete containing the big Y, those end up in the same cell and you end up with that organism being big Y, big Y. Over here, you're showing the result of this gamete being fertilized by this gamete. And so you'd end up with, again, a little y gamete being fertilized by a big Y gamete. Those end up in the same cell. So now you end up with big Y, little y. In the heterozygote, always write the capital letter first. Never put little y, big Y. Okay, that's not the right way to write it. Let's go down here. This offspring right here, or potential offspring, is the result of this gamete being fertilized by this gamete. So again, big Y, little y. And this last box is the result of this gamete being fertilized by this gamete. So that last box would be little y, little y. Look at the phenotypic ratio. So look at the phenotypes. These three potential offspring would all be yellow, and this potential offspring would be green. And so again, Mendel actually did these studies, and he counted up how many green seed producing versus yellow seed producing plants that he got after he did this cross, and he got that same ratio, 3:1. That ratio is going to be really key in lab especially. So the phenotypic ratio would be 3:1. So for every green plant, Mendel got 3 yellow plants. That's the phenotypic ratio. We're talking about color, right? We're saying for every green, there's 3 yellow. The genotypic ratio is a little different. We say the genotypic ratio is 1:2:1. What does that mean? For every homozygous dominant, there's 2 heterozygotes and 1 homozygous recessive. So you can see here, let's circle it. This would be my 1 to 2 to 1 ratio. So that's the genotypic ratio. The phenotypic ratio is the actual colors. Okay, so how do you get the 3:1 ratio? You cross 2 heterozygotes. Big Y, little y, crossed with big Y, little y. That's going to give you a 3:1 ratio. Okay, all the time. So when you think about lab this week, let me erase everything. You're going to have on Cobb 1 and Cobb 3, you're going to see a 3:1 ratio. And we're going to ask you how you got that. And you should come back to this slide and remember how we got 3:1. So for lab, we're looking at kernel color first. The 3:1 ratio is going to be produced by this cross. Okay, 2 heterozygotes being crossed is going to give you a 3-to-1 ratio of purple to yellow in the case of the corn kernels. The same thing when you get to cob 3. This here is going to give you a 3-to-1 ratio, starchy to sweet. Okay? Okay, let's go to the next slide. Let's do a Punnett square together, make sure we understand this. So again, in lab, you're looking at corn kernel color, and if these are the parents here, 2 heterozygotes, we're asking you what the offspring are going to be. So let's set this up. I'm going to take one parent and put its potential gametes here on the left. So if I have a kernel that's big R, little r, okay, so meiosis is going to occur in that plant, and half of the gametes produced in that plant will contain the dominant allele, and half of the gametes produced in the plant will contain the recessive allele. Okay, got to understand meiosis. 4 gametes would be produced, right? But 2 of them would contain the big R, and the other 2 would contain the little r, so they would be the same, right? So we say 50% and 50%. Let's put the other parent up here on the top. So again, big R and little r. So now inside the box, let me grab a different color so we understand the difference. We're going to put the potential offspring. So if this gamete here and this gamete here come together, that's what's going to be in this box. Big R, big R. If this gamete and this gamete come together here, that's going to give us big R, little r, a heterozygote. If this gamete and this gamete come together, I'm going to write that here in this box. Again, another heterozygote. And then lastly, if these two come together, we're going to have little r, little r. So for the genotypic ratio, it would be a 1 homozygous dominant to 2 heterozygotes to 1 homozygous recessive. That would be the genotypic ratio. The phenotypic ratio would be 3-to-1 purple to yellow. Okay? Okay. So what if you have an organism that you know its genotype? So if we're looking at seed color in plants, if you have a yellow seed, you have to ask yourself, how did that little yellow seed get produced? What's its genotype? And hopefully you can say to me, well, yellow is the dominant phenotype. So a yellow seed can either be big Y, or big Y, little y. We're not sure. Could we figure that out? Sure, we can. We can do a cross called a test cross. So again, a test cross is going to be a way to determine whether an organism that has the dominant phenotype, whether that organism is a heterozygote or a homozygote. So is that seed big Y, or is it big Y, little y? How do you do that? You're going to take that unknown organism here, and you're going to cross it with this one. You're going to cross it with a homozygous recessive organism. Okay? And so if you do that, and you get all, in the top one, if you do that and you get all yellow, that's going to be a good indication that your unknown genotype is big Y. Okay? You'd have to have a really large amount of offspring to really say that this is big Y. Okay? The second one is a little bit easier. So in this second one here, again, I'm crossing my unknown with a homozygous recessive. If I get this result, a 1-to-1 ratio of offspring, I know that that unknown parent must have been big Y, little y. Must have been a heterozygote. Because green seeds were produced. Green peas were produced. Okay, so again, you want to make sure you know the definition of a test cross You also, from this slide, are given sorry, I've got my pen here. You are also given on this slide, in the second box, how you get the 1-to-1 ratio. So I want to take a little bit of a closer look at that. And let's look, instead of doing seed color, let's take it back to lab, and let's do this we'll do corn kernel color. So again, kernels can be purple or yellow, and purple is dominant to yellow. So if you have big R, little r, okay, so that would be a purple kernel. Its genotype is big R, little r, or we could say it's a heterozygote. And we cross that plant with a plant that produces yellow kernels, little r, little r. Let's do the Punnett square really quick for this, make sure we understand. So let me put this parent over here on this side. So half of the gametes from this parent are going to be big R, and half are little r. Okay, and let me put this parent, the other parent, at the top. So I'm going to put little r and little r, and let me draw my box. Okay. This gets a little tricky to draw on here. So my first box here is going to end up being big R, little r, heterozygous purple. My second box is also going to be big R, little r. The more practice you have with these Punnett squares, the better you get. My third box down here is going to be little r, homozygous recessive, yellow, kernel producing plants, and the same thing in this last box. And so what do I get? I get 50% purple and 50% yellow, or I could say a 1-to-1 ratio. I could have actually simplified my Punnett square once you get good at these, since if you have two gametes that are the same, you can really get rid of this. And we could have just had a 2-box Punnett square, 50-50 or 1-to-1. So this would be a 1-to-1 ratio of purple to yellow. That's a ratio you're going to see in lab this week. So on cobs 2 and 4, cob 2 is going to show you a 1-to-1 ratio of purple to yellow. And cob 4 is going to show you a 1-to-1 ratio of starchy to sweet. And so when you see the 1-to-1 ratio, you should automatically come back here and know that those are the parents that must have produced that ratio. A 1-to-1 ratio in a monohybrid cross is always produced when you cross a heterozygote with a homozygous recessive individual. Okay. I think I'm actually going to pause the video here. And then you can start the next video. So this is chapter 12, part 1, and I'm going to start the next video because we're just at an hour. Okay, so when you're ready, go ahead and open up the next